This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of one of the most profound shifts in human history, the era of germline engineering. For millennia, we shaped life through observation. Today, we possess the tools to rewrite our own biological code. In this narration, we will trace the evolution of genetic mastery, from ancient selective breeding to the revolutionary precision of CRISPR. We'll examine the high-stakes scandals, the ethical dilemmas of designer babies, and the shadow labs pushing the boundaries of human evolution. Join us as we confront the reality of holding our species' future in our hands. Chapter 1: The Evolution of Genetic Mastery. The ability to intentionally regulate and modify living organisms has been a hallmark of human advancement for thousands of years, long before we understood the existence of a single molecule of DNA. Our ancestors were the first genetic engineers, though they operated through the slow, observational lens of selective breeding. By choosing to propagate only the plants with the largest seeds or the animals with the gentlest temperaments, early societies unintentionally reshaped the genetic variety of the world around them. A classic example of this ancient mastery is the transformation of wild mustard. Through at least 2 millennia of selective breeding for specific features, humans created entirely different-looking cultivars from the same biological root. By selecting for large-leaved plants, they developed kale. By selecting for larger flower buds, they produced cauliflower. This trial-and-error method was effective but vulnerable to environmental risks, disease outbreaks, and the unpredictable whims of nature. The shift from observational breeding to a structured understanding of inheritance began in the mid-19th century with foundational research conducted on pea plants. This work established the basic principles of heredity, describing how traits are passed from one generation to the next through what we now recognize as dominant and recessive alleles. It was the first time humanity glimpsed the underlying rules of the biological game, realizing that characteristics were not just blended randomly, but followed a mathematical logic. However, it took several more decades for science to identify the physical carrier of this information. Between the mid-1940s and the early 1950s, the role of deoxyribonucleic acid, or DNA, as the genetic material became firmly established. The landmark discovery of the double helix structure of DNA in 1953 provided the ultimate stimulus for the development of genetics at the molecular level. For the first time, we could see the software of life, and the race to learn how to edit that software began in earnest. By the late 1960s and early 1970s, molecular genetics began to move from observation to active manipulation. This era was defined by the discovery of a molecular toolkit that included enzymes capable of acting as both glue and scissors. In 1967, researchers isolated DNA ligase, an enzyme that can join 2 strands of DNA together. This was the prerequisite for constructing recombinant DNA molecules, sequences that are not normally joined together in nature. Shortly after, the discovery of restriction endonucleases, more commonly known as restriction enzymes, changed everything. Naturally occurring in bacteria as a defense mechanism against viruses, these enzymes recognize particular sequences of nucleotides and cut the DNA at specific sites. Because they always cut at the same sequence, they allowed scientists to remove a piece of DNA from one organism's chromosome and insert it into another. This led to the birth of gene cloning and the production of the first genetically engineered products, such as human insulin produced by bacteria, which was approved for medical use in the early 1980s. As the toolkit expanded, scientists made the first tentative leaps toward human application. These early human trials represented vital evolutionary steps between the basic tools of the 1970s and the precision nucleases of the 21st century. In 1980, Martin Klein conducted the first, albeit unsuccessful and highly controversial, attempt at human DNA modification. He sought to treat patients with beta thalassemia by inserting functional hemoglobin genes into their bone marrow cells. While the experiment failed to produce a clinical benefit and sparked an ethical firestorm regarding unauthorized human experimentation, it signaled that the era of human genetic engineering had arrived. A decade later, in 1990, French Anderson achieved the first successful nuclear gene transfer in a human patient. By using a viral vector to deliver a functional gene into the white blood cells of a 4-year-old girl with a severe immune deficiency known as AIDA-SID, Anderson proved that genetic material could be successfully integrated into a human host to treat a disease. While these early techniques were revolutionary, they were also remarkably blunt. The primary drawback was the random nature of how new DNA was inserted into a host's genome. Scientists could add a gene, but they couldn't always control where it landed, which meant they might accidentally impair or alter other vital genes. To achieve true genetic mastery, the field needed a way to target specific locations with absolute precision. This led to the development of several generations of engineered nucleases. The first significant attempt at precision was through homologous recombination, a natural process where cells repair damaged DNA by using a similar or identical sequence as a template. By creating DNA constructs that matched a targeted sequence, researchers could trick a cell into swapping out a piece of its own genome for a modified version. While this worked well in simple organisms like yeast and was used to create the first knockout mice, it was extremely inefficient and time-consuming in complex human cells. The quest for better tools led to the development of zinc finger nucleases, or ZFNs, in the early 2000s. These were synthetic proteins designed to bind to specific DNA sequences and create a double-stranded break at a desired location. By linking DNA recognition peptides to a cleavage domain, scientists could finally target almost any point in the genome. However, ZFNs were difficult and expensive to engineer, often requiring months of work to design a tool for a single new target. This was followed by the emergence of TALENs, or transcription activator-like effector nucleases. TALENs represented a significant evolution over ZFNs because they offered a much simpler and more predictable code for DNA recognition. Specifically, TALENs utilize a 1-to-1 recognition ratio between their constituent amino acids and the DNA nucleotides they target. This modularity made them far easier to design and more specific in their binding, yet they remained physically large and difficult to deliver into living cells, particularly the reproductive cells required for germline engineering. The true revolution arrived with the adaptation of a bacterial immune system known as CRISPR-Cas9. Although CRISPR is now synonymous with high-tech editing, its discovery was rooted in natural observation. It was first observed in the genome of E. coli in 1987, appearing as mysterious repeating sequences of DNA. It took nearly 2 decades for researchers to realize that these repeats were not genetic junk, but part of an adaptive immune system. Between 2005 and 2007, scientists identified that these sequences matched the DNA of viruses that had previously attacked the bacteria. CRISPR functions like a molecular library of past infections. If a virus returns, the bacteria produces a guide RNA that matches the viral sequence, leading the Cas9 enzyme directly to the invader to snip its DNA. In 2012, researchers realized they could hijack this system. By designing a custom guide RNA to match any sequence in the human genome, they could direct the Cas9 inches scissors to a specific gene with unprecedented ease, speed, and low cost. The CRISPR system operates through an elegant search and replace mechanism. The guide RNA acts as a GPS, finding a specific sequence among the 3 billion letters of the human genome. Once the Cas9 enzyme makes a cut across both DNA strands, the cell recognizes the damage and attempts to repair it. Scientists can harness 2 main repair pathways. The first is non-homologous end joining, which simply glues the ends back together, often resulting in small errors that effectively disable or knock out a gene. The second, and more powerful for engineering, is homology-directed repair. If a researcher provides a separate template of healthy DNA along with the CRISPR components, the cell will use that template to fix the break, effectively knocking in a new or corrected gene. This technology opened the door to the most controversial application of genetic science, germline engineering. To understand the significance of this shift, we must distinguish between somatic and germline cells. Most genetic therapies today target somatic cells, the muscle, organ, and blood cells that make up the vast majority of our bodies. Changes to these cells affect only the individual patient and die with them. Germline engineering, however, targets reproductive cells, sperm, eggs, or very early embryos. Because these cells provide the genetic blueprint for an entire person, any change made to them is replicated in every cell of the resulting individual's body, including their own reproductive cells. This means the modification becomes a permanent, heritable part of that family's lineage, passed down to all future generations. The scientific community began to explore the potential for using these tools to prevent devastating hereditary diseases, focusing on foundational proof-of-concept targets. One primary focus was the HBB gene, which codes for a protein in red blood cells. Mutations in this gene cause beta thalassemia, a blood disorder that can be fatal without lifelong treatment. By editing the germline to correct these mutations, scientists envisioned a future where such diseases could be eradicated from a family line forever. Another area of intense interest involved the CCR5 gene. The inspiration for targeting this gene was rooted in a remarkable human story, the Berlin patient. Timothy Ray Brown, an HIV-positive man, was functionally cured of the virus after receiving a bone marrow transplant for leukemia. His donor possessed a rare innate resistance to HIV caused by a natural mutation in the CCR5 gene. This real-world evidence of genetic resistance provided a human anchor for the technical discussion of CCR5, suggesting that if scientists could replicate this mutation in embryos, they could confer lifelong viral resistance to future children. As the field matured, researchers developed even more precise variations of the technology to address the risks of traditional CRISPR, such as unintended mutations caused by double-strand breaks. Base editing emerged as a significant advancement, offering a way to make single-letter changes in DNA without cutting the double helix. This is achieved through a chemical mechanism where a deaminase enzyme is fused to a modified Cas9 protein. This allows for the direct conversion of one nucleotide base into another, specifically converting cytosine to thymine, C to T, or guanine to adenine, G to A. By avoiding the physical breakage of the DNA strands, base editing significantly reduces the risk of unintended genomic damage. This was followed by prime editing, which functions like a true search and replace tool, capable of writing new genetic information directly into a targeted site with even greater flexibility. However, the leap from laboratory models to actual human reproduction remained a formidable technical barrier. Making a healthy baby from an edited cell is not just about changing the DNA sequence. It requires the cell to be in the correct epigenomic state. While every cell in our body has the same DNA, they function differently because of epigenetic marks, chemical modifications that tell the cell which genes to turn on or off. This is why a liver cell and a brain cell can have identical genetic software but perform entirely different functions. These marks are crucial for healthy embryonic development. If an engineered cell does not have the right epigenetic instructions, the embryo may fail to develop or result in severe deformities. This epigenomic correctness problem became recognized as a major technical bottleneck, requiring researchers to understand not just the genetic code, but the complex regulatory system that operates on top of it. The transition from the slow, natural process of Mendelian inheritance to the rapid, intentional precision of molecular scissors happened in the span of a single human lifetime. We had moved from observing the rules of nature to possessing the capability to rewrite them. As the tools for editing the human genome became more accessible and powerful, the scientific world found itself at a crossroads, setting the stage for a moment when this speculative science would collide with reality in a way that would shock the global community. If the technical hurdles of precision and epigenomic correctness could be overcome, who would be the first to cross the line from theory to a living, breathing human being? Chapter 2: The Rogue Architect of the Germline. In November 2018, the global scientific community was jolted from its theoretical discussions by a series of YouTube videos that would change the course of human history. A biophysicist named He Jiankui, based at the Southern University of Science and Technology, SUS Tech, announced that he had successfully created the first human babies from genetically edited embryos. These twin girls, known by the pseudonyms Lulu and Nana, were born into a world that was entirely unprepared for their existence. This was not a gradual transition from lab research to clinical application, but a sudden, clandestine leap that obliterated the boundary between what science could do and what society had agreed it should do. The announcement was strategically timed to coincide with the Second International Summit on Human Genome Editing in Hong Kong, ensuring that the news would break just as the world's leading geneticists and bioethicists were gathering to discuss the very ethics He Jiankui had already bypassed. The project that led to the birth of these children was defined by a specific medical goal, conferring resistance to the human immunodeficiency virus, or HIV. To find participants, He Jiankui reached out to the Baihualin BHL China League, an HIV volunteer advocacy group. He focused his recruitment on couples in which the father was HIV positive and the mother was HIV negative. In many parts of the world, including China, such couples face significant social stigma and limited access to traditional fertility treatments. By offering these families in vitro fertilization services, he was able to gain their participation in a project that went far beyond standard reproductive medicine. He Jiankui targeted the CCR5 gene, the viral entry point discussed in Chapter 1, to attempt to replicate the innate HIV resistance seen in rare natural mutations. While the public announcement was framed as a humanitarian breakthrough, the scientific reality revealed by subsequent analysis was a portrait of technical failure and medical risk. The most critical issue identified by experts was evidence of mosaicism in the twins. In Lulu and Nana, the data suggested that the CRISPR components continued to edit the DNA after the embryo had already begun to divide, resulting in a patchwork of cells with different genetic makeups. Some cells carried the intended edit, while others remained unedited or contained entirely different random mutations. This meant that the children were likely not fully resistant to HIV, and the unpredictable interaction between these different cell populations introduced unknown health risks that could persist throughout their lives. Beyond the failures of the editing itself, the experiment was plagued by the threat of off-target effects. In the case of the gene-edited twins, the data indicated that the possibility of unintended mutations was not satisfactorily explored before the embryos were implanted. Unintended cuts in the genome can turn off vital tumor suppressor genes or disrupt other essential biological functions, potentially leading to developmental disorders or cancer. The scientific community was particularly alarmed because these changes are heritable. Any negative consequences introduced by these imprecise edits are now a permanent part of the genetic lineage of these children, capable of being passed down to every future generation they might have. The scandal was further deepened by the revelation of systemic deception and the violation of fundamental research ethics. Investigative reports indicated that He Jiankui had conducted much of the project in secret, even from his own university. To gain approval, he was accused of forging ethical review documents from the Harmony Health Hospital and misleading the reproductive endocrinologists who performed the embryo implantations. These medical professionals reportedly had no idea that they were participating in an unprecedented genetic experiment. They believed they were performing standard IVF. Furthermore, to bypass Chinese regulations that prohibit HIV-positive men from using assisted reproductive technologies, He Jiankui substituted HIV-negative blood samples for the father's actual blood to trick the medical staff into proceeding with the procedure. Perhaps the most damning evidence against the medical necessity of the experiment was the use of sperm washing. This procedure, which removes HIV from the semen, was actually performed during the experiment to prevent transmission during IVF. Because sperm washing already effectively mitigated the primary risk of passing the virus from father to child, the genetic editing was medically unnecessary. The use of a dangerous and unproven genetic modification was viewed not as a life-saving intervention, but as an unnecessary risk taken for the sake of scientific firsts. The global reaction to the announcement was a rare moment of near-universal condemnation. At the Hong Kong summit, the atmosphere was described as one of shock and betrayal. During his presentation, He Jiankui also revealed that a second pregnancy was underway. This third baby, later referred to in reports as Amy, was born in 2019, further cementing the reality that the germline had been irrevocably altered in multiple individuals. Prominent scientists who had previously advocated for a prudent path forward for gene editing found themselves forced to defend the entire field. Critics argued that the experiment was a violation of the implicit social contract between science and society, which relies on transparency and the adherence to shared moral norms to maintain public trust. In the wake of the scandal, He Jiankui was labeled in both scientific and popular press as a rogue agent. However, the massive impact of his work was undeniable. Despite the international outcry, he was listed in Time magazine's 100 most influential people of 2019, illustrating the polarizing nature of his actions. Within China, 122 scientists issued a joint statement strongly condemning the work as unethical and a massive blow to the reputation of Chinese science. They argued that the technology was still too deficient and the risks too uncontrollable for any clinical application in human embryos. The Chinese government, which had initially seen the news reprinted in state media as a historic breakthrough, quickly reversed its stance. Authorities suspended He's research activities, and he was subsequently fired by the Southern University of Science and Technology. The legal fallout was swift and severe. In December 2019, a district court in Shenzhen found He Jiankui guilty of illegal practice of medicine. The court ruled that he and two of his collaborators, Zhang Renli and Chen Jinzhou, had deliberately violated national regulations on scientific research and medical management in a pursuit of personal fame and profit. He was sentenced to 3 years in prison and ordered to pay a fine of 3 million yuan. His collaborators received shorter prison sentences and were banned from working in the field of assisted reproductive technology for the rest of their lives. This trial and the surrounding scandal served as the direct catalyst for a radical shift in the global regulatory landscape. In 2020, the National People's Congress of China passed significant updates to its civil code and criminal law. These amendments explicitly prohibited human gene editing and cloning for reproductive purposes. Crucially, the new law established a much stricter criminal liability for violators, with sentences of up to 7 years in prison for serious cases, a significant escalation from the 3-year sentence He Jiankui received under the previous legal framework. The international response was equally transformative. The World Health Organization, WHO, moved to establish a global registry for all human genome editing research to ensure transparency and accountability, aiming to prevent any future researcher from operating in the shadows. Since the scandal, the WHO has issued 3 separate, comprehensive reports providing guidelines and governance frameworks for human genome editing. These reports emphasize that the technology should only be used for the treatment of disease, and within a framework of rigorous oversight and public deliberation. This moment in history serves as a cautionary tale about the limits of scientific autonomy. It demonstrated that even the most revolutionary tools can be dangerous when wielded without oversight or a clear medical mandate. The scandal did not stop the development of gene editing technology, but it did end the era of self-regulation. It moved the world into a new phase where the development of these tools must be balanced against a rigorous framework of international law and public deliberation. The birth of the first gene-edited humans was not the triumphant milestone its creator envisioned. Instead, it became a global crisis that forced humanity to confront the reality that the power to rewrite our foundational code was no longer a matter of if, but a matter of how we would survive the consequences of doing so. The debate now turned toward the profound philosophical struggle of where to draw the line between the compassionate act of curing a child and the dangerous allure of designing one. Chapter 3: The Moral Boundary of the Human Code. The shadow cast by the first instances of human germline modification has forced a global reckoning with a question that was once confined to the pages of speculative fiction. We are no longer debating a distant possibility, but a present capability. Where is the line between a compassionate medical intervention and the controversial pursuit of human perfection? This tension defines the designer-baby dilemma, a philosophical struggle that pits our desire to alleviate suffering against our fears of a genetically stratified society. At its heart, this dilemma is driven by the dual nature of germline engineering. On one hand, it offers a definitive end to hereditary conditions that have plagued families for generations. On the other, it provides a toolkit for the deliberate selection and enhancement of traits that have nothing to do with disease, potentially rewriting the very definition of what it means to be human. The most compelling argument for germline engineering lies in its therapeutic potential to eradicate monogenic disorders, diseases caused by a mutation in a single gene. For families carrying the genetic markers for conditions like beta thalassemia or Marfan syndrome, the stakes are profoundly personal. Beta thalassemia, caused by mutations in the HBB gene, results in a life-threatening underproduction of hemoglobin, while Marfan syndrome, linked to the FBN1 mutation, can lead to life-threatening complications in the heart and blood vessels. Research has already demonstrated that these mutations can be targeted with remarkable precision. In one study, scientists used base editors to correct an FBN1 mutation in human embryos with a success rate of 16 out of 18 attempts. For a parent who knows they carry such a mutation, the ability to ensure their child is born without it is seen by many as a moral imperative. However, the transition from curing a disease to designing a person is not a clear jump, but a gradual slide down a blurry slope. This is best illustrated by the distinction between therapeutic and non-therapeutic interventions. Consider a hypothetical scenario involving 2 children who are both predicted to grow up to be exceptionally short. The first child has a specific mutation in the gene responsible for human growth hormone. Editing this gene to allow him to reach an average height is widely classified as a therapeutic treatment. The second child is predicted to be just as short, but not because of a single mutation. Rather, he has simply inherited a combination of height-related genes from parents who are also very short. Editing the second child's genome to achieve the same average height is often labeled as an enhancement. In both cases, the outcome is identical, a child of average height, and the impact on the child's quality of life might be the same. The story of Timothy Ray Brown, known as the Berlin patient, serves as a perfect bridge for this debate. While his case proved that a specific mutation in the CCR5 gene could provide resistance to HIV, applying this knowledge to embryos creates a philosophical gray area. Is giving a child innate HIV resistance a cure for a disease they do not yet have, or is it an enhancement of the natural immune system? If we view the lack of resistance as a vulnerability to be fixed, we move toward a world where the absence of an upgrade is treated as a medical deficiency. This ambiguity makes it incredibly difficult for regulators to draw a firm line that allows for the relief of suffering while preventing the rise of cosmetic or performance-based engineering. The concept of the designer baby extends far beyond height or disease resistance. It encompasses the deliberate selection of any trait deemed desirable, from physical strength to cognitive capacity. Animal studies have already provided a proof of concept for these kinds of modifications. In a notable experiment involving Lian Guang's small-spotted pigs, researchers used molecular scissors to precisely edit the myostatin gene. Myostatin naturally functions as a negative regulator of muscle growth. It is essentially the body's internal brake on muscle size. By introducing specific mutations to disable this brake, the researchers produced piglets with significantly increased muscle mass and a higher density of muscle fibers. Because the myostatin gene regulates human muscle growth in a nearly identical fashion, the scientific implication is clear. The same techniques could theoretically be used to engineer humans with exceptional physical strength. The pursuit of cognitive enhancement is even more complex. While traits like height or muscle mass can sometimes be traced to a few key genes, intelligence is a polygenic trait influenced by thousands of small genetic variants. To address these complex traits, the field has turned to polygenic risk scores, which aggregate the estimated effects of thousands of genetic markers to predict a person's likelihood of having a certain trait. The use of these scores for embryo selection represents a different kind of germline engineering, one based on choice rather than direct editing. This practice allows parents to screen multiple embryos and select the one that most closely matches their desired genetic profile. This move toward polygenic screening for non-medical traits has ignited a fierce ethical debate. Critics argue that these predictions often lack clinical validity, as current scores for complex traits like educational attainment only explain a small fraction of the actual variance. Relying on such data to make life-altering decisions for a future child is viewed by many bioethicists as a violation of the child's autonomy, effectively confining their identity within a framework of genetic predictions before they are even born. This leads to the profound question of consent, which remains one of the most difficult philosophical hurdles. Unlike conventional medical treatments where a patient can weigh the risks and benefits for themselves, germline modifications are performed on embryos that cannot consent. Furthermore, because these changes are heritable, the intervention affects an endless line of future individuals. Some ethicists argue that this removes a fundamental right of every human being to a non-manipulated genome. They worry that being a designed person could lead to a crisis of identity, where an individual feels their achievements or traits are not truly their own, but the result of a parental purchase. Countering this view is the principle of procreative beneficence, a framework championed by philosophers like Julian Savulescu. This suggests that parents have a moral obligation to select the child of all the possible children they could have who is expected to have the best life. Proponents argue that we already make countless decisions that shape the lives of our children without their consent, from diet to education. If we have the technology to protect a child from a lifetime of chronic pain or to give them a cognitive foundation that allows for greater flourishing, Savulescu and others argue it would be wrong not to use it. From this perspective, leaving a child's genetics to the natural lottery is not a neutral act, but a choice to accept a potentially lower quality of life for that child. The debate is further complicated by the reality that natural reproduction is itself a form of genetic experimentation. Philosopher David Pearce argues that every time a sperm and an egg meet, the genomes of the parents are scrambled in a process that is entirely untested and often results in spontaneous mutations or the inheritance of debilitating traits. From this viewpoint, germline engineering is simply a more intentional and eventually more controlled version of this natural process. The real question is not whether we should interfere with nature, but how we should analyze the risk-reward ratios. If the risk of a genetic intervention is lower than the risk of a natural genetic disorder, the intervention becomes the more ethical choice. However, a vital philosophical counterpoint to procreative beneficence is the value of disability and human diversity. Many individuals with genetic conditions argue that their conditions are not defects to be fixed, but are integral parts of their identity and unique perspective on the world. They fear that a widespread push for germline engineering will lead to a culture of intolerance for human variation. If society begins to view certain traits as errors to be edited out, it risks losing the diverse range of human experiences that have historically enriched our culture. This perspective challenges the assumption that there is a correct or optimal human genome. The risk side of the ethical ratio is also weighed down by the problem of epigenomic correctness, where the failure to maintain the proper regulatory state of a manipulated cell can lead to catastrophic developmental failure. Beyond this technical hurdle, the potential for unintended consequences looms large. In our current state of knowledge, the genome is a highly integrated system that we only partially understand. A gene that is associated with a desirable trait in one context might have hidden negative effects in another, a phenomenon known as antagonistic pleiotropy. For example, a genetic variant that provides a boost to the immune system might also increase the risk of autoimmune disorders. Because germline changes are permanent, any mistake is not just a personal tragedy for one individual, but a permanent alteration to the human gene pool. Public opinion on these issues reflects a deep-seated caution. Surveys, such as those conducted by the Royal Society in the United Kingdom, show a significant divide in support based on the intended use of the technology. While a large majority, around 76%, of participants supported the use of germline engineering to prevent or correct life-threatening diseases, that support dropped precipitously for non-medical enhancements. Only about one-third of respondents felt it was acceptable to use these tools to alter traits like eye color or to enhance intelligence. This suggests a broad societal consensus that while the relief of suffering is a noble goal, the pursuit of a perfect human is viewed with profound suspicion. Religious perspectives on the dilemma provide distinct frameworks for navigating these boundaries. In Judaism, there is a strong emphasis on the duty to heal and reduce suffering, often leading to a more supportive view of therapeutic germline interventions as an extension of the mandate to preserve life. Conversely, many Christian traditions, particularly within Catholicism, express deep caution regarding playing God and the potential for the commodification of human life, emphasizing the sanctity of the natural reproductive process. Islamic perspectives generally accept therapeutic uses that restore health, but are wary of enhancements that are seen as changing God's creation for the sake of vanity or superiority. Buddhism often focuses on the intention behind the technology, suggesting that if the goal is the alleviation of suffering and the promotion of well-being, the tools can be used ethically, provided they do not reinforce the ego or create new forms of attachment and suffering. These diverse views highlight that the struggle to define the boundaries of germline engineering is not just a technical challenge, but a struggle to define our own values. As we stand on the threshold of being able to rewrite our foundational code, we are forced to ask whether our primary responsibility is to the individual child, to the future of the species, or to a natural order that has governed life for eons. The dilemma of the designer baby is ultimately a mirror reflecting our deepest hopes and our most profound anxieties about what it means to be human in an age of total biological control. If we choose to step across this line, can we ever truly know the cost of the perfection we seek? Chapter 4: The Genetic Divide and the New Eugenics. The historical shadow of eugenics has long loomed over the field of genetics, but the emergence of germline engineering has transformed this specter from a state-mandated nightmare into a market-driven reality. In the early 20th century, eugenics was characterized by coercive government policies, including marriage restrictions and selective sterilizations, aimed at improving the collective genetic inheritance of a population. This history remains a significant blemish on Western democracies, particularly in the United States, where eugenicist policies were once codified into law and remain a largely unaddressed chapter of the national narrative. Today, we are witnessing the rise of what some call techno-eugenics or liberal eugenics. This new iteration is not driven by the dictates of a central authority, but by the private choices of individual parents operating within a competitive global economy. While the motivation is often the well-being of a future child, the aggregate effect of these private decisions could lead to a fundamental restructuring of human society, creating a world where biological destiny is a commodity purchased by the few. The primary engine of this potential divide is the staggering cost of genomic medicine. We can look at existing somatic gene therapies to understand the economic barriers that will likely define the first generation of germline interventions. For example, the first approved CRISPR-based therapy for sickle cell disease represents a functional cure for a devastating condition, yet its price tag is expected to exceed $1 million. Other genomic medicines are already costing upwards of $2 million per patient. In many wealthy Western countries, these treatments are frequently denied coverage by public health bodies and private insurers, effectively restricting them to the extremely affluent. If these are the costs for treating a single individual after birth, the specialized laboratory procedures required for germline engineering, including in vitro fertilization, embryo culture, and high-precision editing, will almost certainly be even more exclusive. This economic barrier creates the risk of a biological caste system. In our current society, wealth already provides significant advantages in terms of nutrition, education, and healthcare. But germline engineering introduces the possibility of positional goods that are hard-coded into the DNA. Building on the traits like height and cognitive capacity discussed in previous chapters, these modifications provide value primarily because others do not have them. However, the pursuit of these traits through polygenic scores introduces the risk of good-hearting, a concept where a measure ceases to be a good measure once it becomes a target. If parents begin optimizing embryos to achieve a specific genetic score for intelligence or stature, the score itself may lose its relationship to the actual trait, potentially leading to unintended biological consequences while further entrenching social hierarchies. We face a future where the gap between the haves and the have-nots is no longer just a matter of bank accounts, but a matter of heritable biological advantage. The potential for this divide is not limited to individuals within a single nation. It extends to a global scale. Current data shows that approximately 90% of all gene-editing clinical trials are concentrated in high-income countries. This geographic imbalance means that the benefits of these technologies are being developed for and tested on a narrow demographic, predominantly of Caucasian ancestry. This lack of genomic diversity in research means that tools and reagents may be less effective or even dangerous for populations with different genetic backgrounds, who might carry variants that make them more susceptible to off-target effects. As high-income nations forge ahead with these capabilities, the rest of the world risks being left behind in a state of genetic underdevelopment, unable to access the tools that could reduce their national disease burdens. To address these mounting ethical and equity concerns, scholars have established the Global Observatory for Genome Editing. Led by figures such as Sheila Jasanoff and Krishanu Saha, this initiative seeks to facilitate a more inclusive international conversation that moves beyond the narrow technical questions of safety and efficacy. The observatory aims to challenge the assumption that the development of these technologies is an inevitable march of progress, instead asking how they might be governed to serve the global public good rather than just the interests of the wealthy few. The issue of corporate control and intellectual property further complicates this landscape. The foundational tools of the gene editing revolution are currently the subject of intense legal disputes, most notably the high-stakes patent battle between the University of California, Berkeley and the Broad Institute of MIT and Harvard. Corporations and research institutions are fighting for exclusive rights over the ability to edit the code of life. If a few powerful entities successfully monopolize these techniques, they can use patent law to restrict access and keep prices artificially high. This commercialization of the human genome turns our collective genetic heritage into a proprietary asset. When the ability to prevent a child's suffering or enhance their potential is locked behind a corporate paywall, the principles of justice and equity are profoundly undermined. This market-driven approach to human traits also raises concerns about the stigmatization of the unmodified. In a world where certain genetic conditions can be edited out before birth, society's commitment to supporting and accommodating people with those conditions may begin to erode. Many individuals living with genetic conditions worry that a widespread push for germline engineering will lead to a culture of intolerance for human variation. If we begin to view certain traits as defects to be eliminated, we risk losing the diverse range of human experiences and perspectives that have historically enriched our culture. Beyond the social implications, there is a serious population-level risk related to the loss of genetic variation. Evolution has equipped the human species with a vast library of genetic diversity, which serves as a critical buffer against environmental changes and new pathogens. Some traits that we currently view as undesirable may actually provide hidden advantages. A well-known example is the sickle cell polymorphism, which, while causing disease in its homozygous form, provides a survival advantage against malaria in its heterozygous form. If we use germline engineering to systematically uniformize the human gene pool according to our current limited understanding of what is desirable, we may be inadvertently stripping our species of the resilience needed to survive future biological threats. The commodification of offspring is also visible in the emerging practice of selecting savior siblings. Through pre-implantation genetic diagnosis, parents can screen embryos to find a child who is a perfect genetic match for an existing sibling who needs a life-saving transplant. While this is done out of a desperate desire to save a child, it treats the new baby, at least in part, as a biological resource rather than an individual with their own autonomous ends. This shift in the parent-child relationship from one of unconditional acceptance to one of conditional selection based on utility parallels the broader trend of viewing children as projects to be optimized. Recognizing the need for a broad societal debate that includes more than just scientists and billionaires, some nations have experimented with citizens' juries on genome editing. In Australia, and through a Dutch consortium, these juries have brought together diverse groups of laypeople to deliberate on the conditions under which germline editing might be acceptable. These real-world attempts at democratic oversight suggest that when given the opportunity, the public often favors a much more cautious and equity-focused approach than the commercial sector. Even the prospect of making these technologies inexpensive does not fully resolve the ethical dilemma. Some argue that if germline engineering were as cheap and universal as a vaccine, the inequality concerns would vanish. However, this ignores the competitive nature of many human traits. This creates an enhancement treadmill where parents feel forced to genetically modify their children just to ensure they aren't left at a competitive disadvantage. In such a world, the choice to remain unmodified becomes a choice to accept a lower social and economic status for one's child, making the technology effectively coercive even without a government mandate. As these technologies move from the laboratory to the commercial sector, the pressure to implement them is growing, increasingly fueled by immense private wealth. High-profile figures in the technology industry, including Coinbase CEO Brian Armstrong and OpenAI CEO Sam Altman, have already begun underwriting startups like Preventive that aim to bring germline interventions to market. This influx of private capital from individuals who often view biological limits as technical problems to be solved is shifting the momentum away from public oversight and toward a private, accelerated timeline. The involvement of such influential backers suggests that the future of the human genome may be shaped as much by the philosophies of Silicon Valley as by the deliberations of bioethicists. Can a global consensus on the public good survive the sheer financial and ideological force of those who see the human code as the next great frontier for disruption? Chapter 5: Shadow Labs and the Unstoppable Human Redesign. The history of science is often viewed as a slow and methodical climb, but the reality of human germline engineering is increasingly resembling a high-speed train that has already left the station. While international summits and bioethical committees continue to debate the theoretical boundaries of what should be allowed, a new and highly motivated group of actors has entered the arena. These are the private commercial ventures, often operating in the shadows of the traditional scientific establishment, backed by the immense wealth of Silicon Valley and driven by a philosophy that views biological limits as just another set of bugs to be patched. This shift from the public university lab to the private startup incubator has created a new set of challenges for global regulators, who find themselves trying to police a technology that is increasingly well-funded, mobile, and difficult to track. One of the most striking examples of this new frontier is the report of a clandestine startup named Preventive. Led by CEO Lucas Harrington, the company has reportedly been working quietly to usher in the first known birth of a genetically modified baby outside of China. The project is focused on a specific and humanitarian goal: erasing a hereditary disease from a family line. The company has allegedly identified a couple who are interested in these services, though the public face of the venture has denied that such talks are taking place. What makes this effort particularly significant is its backing. The startup is underwritten by some of the most influential figures in the modern technology industry, including the chief executives of major cryptocurrency and artificial intelligence firms. This represents a fundamental shift in the power dynamics of genetic research. When the resources of a small nation are concentrated in the hands of a few private individuals who believe in the moral necessity of their work, the traditional levers of government control, such as the withholding of federal grants, lose their effectiveness. The regulatory environment in the United States currently exists in a state of curious paradox. On one hand, an act of Congress strictly prohibits the Food and Drug Administration from even considering applications for human trials that involve germline gene hacking. This effectively bans the commercial path to a gene-edited baby within American borders. However, this congressional ban only applies to research conducted with federal funds. This means that a strictly private commercial venture, using its own money and resources, is technically operating in a legal gray area. While any scientist caught performing such work would likely become a pariah in the mainstream academic community, the threat of social exile is a weak deterrent for those who are already part of a counterculture that celebrates the idea of the maverick innovator. These startups are often compelled to move their clinical operations outside the United States, seeking out jurisdictions with more permissive regulations or less stringent enforcement, a practice known as jurisdictional arbitrage. Preventive is not alone in this pursuit. A growing number of startups, with names that evoke the ambitious scientific projects of the past, like Manhattan Project and Bootstrap Bio, are entering the arena. These companies argue for a transparent, if aggressive, path toward making heritable editing a reality. They are often supported by a movement known as pronatalism, which advocates for increased birth rates and the use of technology to ensure that future generations are as healthy and capable as possible. These organizations operate with a move fast and break things ethos that is fundamentally at odds with the precautionary principle favored by the international bioethics community. They view the current global moratorium on germline engineering not as a necessary safeguard, but as a temporary hurdle that will eventually be cleared by the sheer momentum of technical progress. Even as direct editing remains a high-stakes gamble, other companies are finding ways to implement a softer form of germline engineering through selection. A company named Nucleus has entered the market offering polygenic screening at a price point of just under $10,000. This service allows parents to look at the estimated effects of thousands of genetic variants to predict a person's risk for certain conditions or traits. Another venture, Heresight, has made even bolder claims, suggesting it can predict an embryo's IQ. While many geneticists argue that these predictions are statistically shaky and lack clinical validity, the existence of a commercial market for this data shows that the demand for designer traits is already being met. These selection-based methods serve as a gateway, normalizing the idea that a child's genetic makeup is a set of data points that can and should be optimized before birth. The technical reality of the field suggests that the bottleneck to truly strong germline engineering is not the ability to sequence a genome or even the ability to edit a few genes. Instead, the primary barrier is a phenomenon known as epigenomic correctness. For an engineered cell to successfully grow into a healthy baby, it must possess the correct epigenomic state. This means the chemical modifications on the DNA, which control how genes are expressed, must match the highly specialized state of a natural sperm or egg. If an embryo is created from a cell that has been heavily manipulated in a lab, it often lacks this epigenomic competence, leading to developmental failure or severe abnormalities. Current research is focused on creating an atlas of human embryonic development at the single-cell level to understand these instructions. Scientists are also exploring ways to grow human eggs in the lab through ovarian follicle culture, a technique that could provide a steady supply of cells for research and eventually for clinical use. Another promising but difficult path forward involves the concept of intact chromosome transplantation. This method would theoretically allow researchers to move entire chromosomes from a donor cell into an embryo, bypassing the need for the hundreds of individual edits required to modify complex traits. While this sounds simple in theory, the mechanical challenges are immense. Using a micromanipulator to move a chromosome is like trying to move a single hair with a pair of bulldozer claws, and more automated methods like flow cytometry often break the fragile DNA. Despite these hurdles, some researchers believe that chromosome selection could eventually become a dominant method because it might bypass the epigenomic correction problem entirely, as the chromosomes would be taken from cells that are already in a stable state. The global landscape for this research is a patchwork of conflicting laws and guidelines. As of late 2020, 96 countries had established policies regarding the use of germline engineering. 70 of those nations explicitly prohibit the use of the technology for reproduction, while others, like Italy, Panama, and the United Arab Emirates, allow it only under very specific exceptions. China, once seen as the Wild West of genetic research, has implemented some of the world's strictest criminal penalties in the wake of recent scandals, with the potential for 7-year prison sentences for those who perform unauthorized germline edits. Meanwhile, the World Health Organization is attempting to maintain a global registry to track this research, but the registry is voluntary and relies on the honesty of the researchers themselves. This lack of a binding international treaty means that there is no global enforcement mechanism to stop a well-funded private group from proceeding in secret. To bridge the gap between speculative science and safe clinical application, some experts have called for the creation of dedicated research centers focused on non-human primates. Because monkeys share a high degree of physiological and genetic similarity with humans, they are the essential model for verifying that these technologies can produce healthy offspring without unintended consequences. Such centers would be tasked with creating an atlas of single-cell RNA sequencing data to map out the natural range of variation in the epigenome. This basic science is the unglamorous but necessary foundation that must be laid before any responsible move toward human trials can be made.
Without this data, any attempt to create a gene-edited human remains a leap into the dark.
The field of germline engineering is often described by those within it as an enchanted field. This term refers to the fact that many of the component technologies are ripening simultaneously, and an advancement in one area, such as a more efficient way to culture cells, can suddenly unlock the potential of another area, like CRISPR precision. This synergy creates an innovation ramp where the power of the technology can grow exponentially. We might start with the correction of a single devastating mutation for a few rare diseases, but as the costs fall and the precision increases, the pressure to move toward more complex traits like disease resistance or cognitive capacity will become nearly irresistible. The rise of these private commercial efforts ensures that the technology will continue to advance regardless of the public debate, as the desire of parents to give their children every possible advantage is a force that few regulations can truly contain. The commercialization of this technology also brings the challenge of scaling. For germline engineering to be socially beneficial rather than a source of further inequality, it must be made inexpensive and widely accessible. This requires not just scientific breakthroughs, but a fundamental rethinking of how we manage intellectual property in the life sciences. The current environment of high-priced genomic medicines and intense patent litigation over CRISPR components suggests that the first generation of these tools will be luxury items. If the ability to edit the human germline remains a proprietary asset of a few corporations or individuals, the risk of a permanent genetic divide becomes a mathematical certainty. The challenge for future policymakers is to find a way to encourage the innovation coming from the private sector while ensuring that the foundational code of the human species does not become a closed-source product. As these startups continue to push the boundaries, the role of the individual scientist is also changing. The traditional model of the academic researcher, motivated by peer review and public service, is being supplemented by the entrepreneur scientist, who is motivated by a vision of a post-human future. This shift has led to a breakdown in the consensus that once governed the field. Some leading researchers now argue that the ethics are actually holding back scientific progress, and that the potential to reduce the global burden of disease is so great that it is immoral to wait. They suggest that the risks of inaction, the continued birth of children with devastating genetic conditions, are higher than the risks of proceeding with carefully monitored trials. This perspective views the unstoppable train of genetic engineering not as a threat to be stopped, but as a vehicle for human liberation that must be steered towards its most beneficial destination. The technical roadmap for the next 2 decades includes several key milestones that will determine the feasibility of these goals. These include the development of methods for the transplantation of spermatogonial stem cells, which could allow a father to produce mutation-free sperm, and the refinement of base editing to a point where off-target effects are virtually non-existent. Each of these steps moves us closer to a world where the natural lottery of reproduction is replaced by the intentional design of the germline. The rise of clandestine startups is simply the first visible sign that the era of theoretical debate is ending and the era of practical implementation has begun. The challenge now is to build a regulatory framework that is as dynamic and global as the technology it seeks to govern, acknowledging that the power to rewrite our foundational code is a reality that cannot be unlearned. The focus of the scientific community is shifting from whether we can perform these modifications to how we can verify the genomic and epigenomic competence of every cell used to create a new human life.
Germline engineering has evolved from selective breeding to precise CRISPR tools, offering cures for hereditary diseases but raising profound ethical questions. The He-Gen-Kwai scandal revealed the risks of unchecked experimentation, while broader implications include genetic divides and the commodification of life. As technology advances, we must choose. Will we edit our genome to heal or to design a divided future? This power challenges our very definition of humanity. Thank you for listening.